Hi everyone, my name is Trisanna. I am from the Botrytis Lab at the Montreal Neurological Institute here in Canada. And today I'm going to be speaking to you as part of our symposium entitled Mapping Cortical Morphology in the Human and Non-Human Primate Brain and Relations to Function. And I'm going to be talking to you about studies from our lab investigating sulcal morphology in the human brain and relations to specific aspects of functional processing. So perhaps you're wondering, why should we care about cortical morphology? Certainly the sulci and gyri form very complex patterns. They seem to be quite variable from one individual to the next. We know that the cerebral cortex has expanded immensely over evolution. This is illustrated by this beautiful figure here of Katja, Heuer, and colleagues. And in fact, the cortex of the human brain is the most convoluted of any primate brain. Furthermore, we know that about 2/3 of the cortex is hidden within its sulci and fissures. And so when you're looking at the lateral surface or the medial surface of the hemisphere, you actually can't see quite a lot of cortex that is hidden within those folds. And we want to be able to map that cortex properly. Finally, as anatomists such as Brodmann demonstrated, and these are his maps shown here, certain sulci and gyri can serve as excellent morphological landmarks for underlying cytoarchitectonic areas that have specific functions. So for example, on the anterior bank of the central sulcus, you are always going to find area 4, which is your primary motor cortex. On the posterior bank, you are always going to find area 3, which is your primary somatosensory cortex. On Heschl's gyrus, hidden within the sylvian fissure, you will find area 41, which is your primary auditory cortex. And within the calcarine sulcus, which is a medial sulcus here, you are going to find area 17, which is your primary visual cortex. And the general belief has been that these relationships hold for the primary sulci, which are the first to form in utero and which are the most consistent across species, but that these relationships don't really hold for the so-called higher level cortical areas. And so our lab is interested in investigating such relationships. In terms of mapping cortical folds, it really became possible towards the end of the 19th century when we could harden brain specimens using alcohol and then actually perform dissections and open up cortical folds and examine the morphology. And these early studies, such as those of Eberstaller in 1890 or Retzius, even Economo and Koskinas, demonstrated that there are relatively consistent patterns across individuals and that there are relations with functional processes, as I just mentioned. However, these studies would have been performed on postmortem specimens, which would have meant a restriction in sample size and also a restriction mainly to the cortical surface. With the advent of neuroimaging tools, we've been able to study lots and lots of subjects who are alive and study the cortical folds in 3 dimensions. And so that's just what I'm showing you in this image on the right here. You can label specific sulci in 3D. You can follow a sulcus from the cortical surface to within the depth of the hemisphere. And you can even look at coordinates in a stereotaxic space, such as that of the Montreal Neurological Institute. And our lab has contributed extensively to this type of research. These are just some of the articles that have come out of our laboratory investigating the morphology of specific sulci. We've also published 3 atlases on this topic, and this is the most recent lateral schematic to emerge from the lab. And actually, we've named some of the sulci for the first time in our lab. So this was actually the basis of the research of my PhD. So I looked at the morphology of the sulci in the ventrolateral frontal cortex. This forms Broca's area for language production in the language dominant hemisphere. I looked at the anterior ascending ramus of the lateral fissure, the horizontal ascending ramus of the lateral fissure, and this occasional sulcus called the sulcus diagonalis. And specifically, I looked at the patterns of these sulci, what is the frequency across hemispheres and individuals, and I then quantified the variability and extent of these sulci using spatial probability maps. So these are the surface probability maps of some of those sulci shown here. So armed with a more extensive understanding of sulcal morphology in our lab, we've been able to examine relations to function using fMRI. And more importantly, we do so by analyzing the data at the individual subject level. So we take into account the individual differences in cortical morphology in relation to function. And these are some of the studies that have investigated anatomal functional relationships using such methods. And I'm going to go into a little bit of detail about 4 of these today. So to start, we have the research of Veronika Zlatkina, who was a PhD student in the lab. She's now a research associate in the lab. And she was very interested in the morphology of the post-central sulcus. We know from the pioneering work of Penfield and Rasmussen and other colleagues that the post-central gyrus is somatotopically organized. This is the famous somatosensory homunculus of Penfield here. And so Veronika was interested in how the post-central sulcus relates to sensory motor function. So in one study, she examined in vivo MRI, and she was able to demonstrate that the post-central sulcus can be subdivided consistently into 5 different segments, as you can see here. And she then examined how these 5 segments relate to specific aspects of sensory motor function. So she had a separate set of subjects enter the scanner, and they had to move different parts of their body depending on the instructions that appeared on the screen. So move the foot, the leg, the arm, the hand, the mouth, the tongue, or blinking of the eyes, all while fixating. And as the control task, it was simple fixation. And she was able to demonstrate that there was a tight relationship between sensory motor representations at different parts of the body and specific segments of the post-central sulcus. You can see that shown really nicely, for example, in this image here. More recently, we've just submitted this work to cerebral cortex. It's under review. Veronika quantified the spatial variability and extent of each of these segments of the post-central sulcus using volumetric and surface probability maps. So these are the maps, for example, of segment 1 that I'm showing you here. And she then looked at how each of these maps relates to sensory motor activation peaks that had been reported in the literature. And that's what this image on the right is showing you. So basically, she was able to reproduce what she had previously demonstrated, namely that each segment of the post-central sulcus could be reliably related to specific sensory motor activation peaks. And the more dorsal segments, for example, were related to sensory motor activation peaks of the lower extremities, the foot, the leg, while the most ventral segments were related to activation peaks of the mouth, the lips, and the tongue. Moving more anteriorly into the dorsal premotor region, we have the study of Celine Amiez. So she was a postdoctoral researcher in the lab, and of course she's going to be presenting her own research as part of this symposium. So separate set of research. But Celine was interested in activity generated during visual motor hand-arm conditional responses and activity related to the frontal eye fields. And the reason for this is that in the monkey, these are quite separate areas. This has been established by lesion studies. It's been established by electrophysiological recordings in the monkey. But in the human neuroimaging literature, these areas seem to consistently have overlapping coordinates at the junction between the superior frontal sulcus and the superior precentral sulcus. And so Celine wanted to investigate this by looking at individual morphology in relation to the functional peaks. So she put a group of subjects into the scanner. And in the first experimental task, these subjects had to press specific keys on a keyboard with a specific finger, depending on the color that appeared on the screen. So this is what we mean by a visuomotor hand conditional response. It was dependent on the color that appeared on the screen. And as the control task, the subjects would simply press the same key, always with the index finger, no matter what color appeared on the screen. As a second experimental task, in order to activate those voluntary saccades, she had subjects follow a dot on a screen for a certain amount of time. And then as the control task, it was a simple fixation. And so Celine was able, by studying the subjects at the individual subject level, to demonstrate that despite considerable variability in local sulcal morphology, there were consistent relationships between anatomy and function. So activity related to the visuomotor hand conditional responses was always associated with the dorsal branch of the superior precentral sulcus, which is shown as the blue peaks in some of these subjects that I'm showing you here. And activity related to the frontal eye fields was consistently located at the ventral branch of the superior precentral sulcus, so those red peaks in this image here. So she was able to demonstrate that local morphology could predict functional organization in this region. And furthermore, that the organization of the dorsal premotor cortex was actually preserved from monkey to human. And so this is an average of the data across subjects. These crosses here represent the average coordinates, and you could see the organization of the average functional peaks here. And it really does reproduce what we know in the monkey, where each of these areas can be reliably separated in the dorsal premotor cortex. Moving on to the medial side of the hemisphere, we have the work of Sonja Handgebut, who was interested specifically in the parahippocampal gyrus. So we know from the work of Brenda Milner with patient HM and also many other researchers that the parahippocampal gyrus is important for consolidation of certain types of memory. And research has demonstrated, especially in neuroimaging, the potential existence of a parahippocampal place area, a region in the posterior parahippocampal cortex that seems to be active to visualization of scenes or locations as opposed to objects or faces. And so Sonja was very interested in how the cortex, specifically the entorhinal cortex, which lies on the gyrus more anteriorly, and the parahippocampal cortex more posteriorly, what is the role of this cortex in spatial memory? So as a first study, she examined the intricate patterns of the sulci in this region. So specifically the rhinal sulcus anteriorly, shown in yellow here, and the collateral sulcus proper, shown in blue here. And once she understood the morphology, she could then go and study relationships to function. So she had 14 healthy subjects who had to learn the layout of a virtual city just by navigating this virtual city on a computer screen. And you could see that this city had different landmarks in various locations. And so they would learn the layout of this virtual city through exploration, and they had to really consolidate that information. And then once they had learned the layout, they headed into the scanner and they were given a random starting point, such as the bank shown here, and they had to navigate to some other landmark, like the church, using the shortest distance. So they really had to retrieve that cognitive map that they had established. As a control task, the subjects navigated a very similar city that was missing these these landmarks, and they would just follow arrows that were put in front of them. So really not relying on that cognitive map. So Sonja was able to demonstrate that the sulcal morphology of this region could be consistently used to describe the location of the activation peaks that had been generated. So she had noticed one cluster of peaks that was generated where the anterior branch of the collateral sulcus proper meets the posterior branch. There was a second cluster of activation peaks generated at the posterior collateral sulcus proper, just where it starts to meet with the lingual gyrus. And a third cluster of peaks that was generated on the parahippocampal extension of the collateral sulcus. And so in this study, she was able to demonstrate specific activation in the mid and posterior parahippocampal cortex. So not the anterior parahippocampal cortex, not the entorhinal cortex. And furthermore, these studies established morphological landmarks that could be related to these activation peaks. we have the study of Emily Siegel, who finished her PhD in the lab several years ago. Emily was very interested in the morphology of the angular gyrus and its role of this region in reading activity. So we know from patient studies, from neuroimaging studies, that the angular gyrus in the language-dominant hemisphere does play a role in reading functions. And Emily had studied the morphology of the superior temporal sulcus and demonstrated that it can be consistently subdivided into 3 posterior branches that really define the angular gyrus of the parietal lobe. So Emily was then interested in figuring out how does the morphology of this region and these 3 branches relate to reading function. And so she had 9 subjects who entered the scanner. They had to view a word on a screen. They had to silently read the word and then write down that word. And as a control task, the subjects would view a picture of an object on a screen and write down the name of the object. And by subtracting these 2 tasks, Emily was trying to hone in on silent word reading. And what Emily was able to demonstrate, and you can see these are the individual subjects, the anatomical and the functional data overlaid, and this is an average of the subject peaks shown here on the left, was that there was a consistent relationship between reading-related activity and the location of the central and the posterior branches of the caudal STS in every single subject. So in conclusion, we don't have much time left to go into even more detail, but thanks to advances in neuroimaging, we have a much better understanding of cortical morphology compared to the original studies of Eberstaller, 1890, and colleagues. We've come quite a long way since that pioneering work that really set the stage for this research. And by examining sulcal morphology at the individual subject level in relation to functional activations, we have been able to establish specific morphological landmarks that can be used for navigating the brain. And you can imagine that this would be very useful, for example, during neurosurgery, during tissue resection of an epileptic focus or of a tumor. If you understand these anatomo-functional relationships, it can be quite beneficial. So with that, I would like to thank the Petrides Lab and especially past and present members whose research I discussed today. I'd like to thank the OHBM committee for selecting our symposium for this year's conference. I would like to thank the other symposium speakers who you'll hear from, Celine Amiez, Olivier Coulon, and Katja Heuer. I'd also like to thank Nicole Eichert for helping me to organize this symposium. And finally, I would like to thank you for your attention.
Hi, I'm Celine Amiez, and today I'm going to present you some recent data that I've got showing that the sulcal organization in the medial frontal cortex provides insights into primate brain evolution. So why studying sulci? To my point of view, there is 3 main reasons. The first one is that the organization of sulci is absolutely not arbitrary. If we look at, for example, here, what is the topic of interest of the present talk, that is the medial wall, you can see here that it's characterized mainly by 2 sulci, the cingulate sulcus that is there in all hemispheres, and the paracingulate sulcus here in pink that is there only in about 70% of subjects, at least in one hemisphere. It's a secondary sulcus occurring later in utero. And if you look at what's going on in the macaque, you can see also that this cingulate sulcus is present in all hemispheres. The second reason is that all my work from the past 15 years converge toward the fact that sulci are pertinent to function. And I show you here one specific example of this region. We have seen that this region that is located at the intersection with the posterior vertical paracingulate sulcus here and the paracingulate sulcus if present, or with the cingulate sulcus if the paracingulate sulcus is absent, contains the face motor representation of the cingulate motor area, the anterior one. So you can access this by with fMRI, asking the subject to perform face movement, like eye movement or tongue movement. And this is also the locus of activity of the region involved in the analysis of visual and Jewish feedback, specifically in exploratory situation. So it's when you are in a trial and error learning, you have to interpret the feedback that you've get, that you've got to adjust appropriately your behavior in the next trials. So this region is involved in the analysis of visual and Joyce feedback in this specific context. And this is interesting to have a look on what's going on in macaque, where we know that this region that seems to correspond, anatomically speaking, is also, you also find here feedback-related activity, Joyce feedback-related activity. So this is the dorsal bank of the cingulate sulcus. This is a meta-analysis of electrophysiological and anatomical studies. And here is the frequency, the more reddish, the higher papers showing activity, feedback-related activity in this region here. And you can see that this region here coincide with this one here that correspond to the eye face motor representation. So you have to imagine this line within the dorsal bank of the cingulate sulcus here. So this means that the functional organization of the mid-cingulate cortex can be predicted from local morphology in human and is well preserved from macaque to human. And the third reason for assessing sulci is the goal of my talk today is to show you that the organization of sulci may be used as a tool to understand primate brain evolution. And to assess this, we analyzed the anatomical T1 MRI data of 197 human brain from the Human Connectome project, 225 chimpanzee brain from collaboration with Bill Hopkins in US, 88 baboons brain thanks to a collaboration with Adrien Megardici in France, and 80 rhesus macaques from our lab and our collaborators. If we look at now what's going on in the dorsomedial frontal cortex, this blue region here, the first thing that we have seen is that the paracingulate sulcus, which was thought to be a human-specific sulcus, is in fact not, because we observe this sulcus also in chimpanzee, as you can observe here. Two main differences can be, however, observed. First, in human, we observe this sulcus in about 70% of subjects at least in one hemisphere. And this percentage falls to 34% in chimpanzee. And the second one is that it's lateralized on the left hemisphere in human, but it's equally present in the left and right hemisphere in chimpanzee. So now if you look at the vertical sulci that are present in this region, we can see that they are present as sulci in both human and chimp. And in baboon and macaque, we observe the precursors of sulci that we call spur or dimple, depending on the shape of it. And if we look at the occurrence of these vertical sulci in all species, we can see that the paracentral sulcus here, the most posterior one, is almost equally present in human and chimp, and equally present in baboon and macaque, but the frequency of occurrence is lower in old world monkeys than in hominids. And if we look at now the morphology of these precursor of sulci or sulci, you can see that in human and chimp, they are all sulci. In baboon, they are most often the spurs than dimple, and most often dimple than spur in macaque. If we look at now the pre-paracentral sulcus that is there, you can see that it's again equally present in human and chimp, and equally present in baboon and macaque, but with a difference between these hominids and old world monkeys' brain. We observe the same morphology pattern with the fact that this sulcus is a sulcus in human and chimp, and a spur in baboon, and a dimple in macaque. The posterior vertical paracingulate sulcus here is it's almost there in all hemispheres in human. It falls a bit in chimp, and it's less there in baboon and macaque. And the morphology, we can see that it's similar to the other sulci. And the anterior vertical paracingulate sulcus here, we can see a linear decrease of the frequency of occurrence across these four species, and the same morphological feature. Now, if we look at so this means that macaques do possess all the precursor of the human vertical sulci in the dorsomedial frontal cortex. And now if we look at where they are localized, this vertical sulci, it was really interesting because we saw that the paracentral sulcus is always found at the level of the rostral limit of the pons here. So This is observed when you have orientate the brain so that the anterior commissure, posterior commissure line is horizontal, and you are in template. That is the MNI template in human, the chimpanzee template in chimpanzee. In baboon, it's a template developed by Adrien Magadichian, and in macaque, it's the MNI template here. So here is the density of occurrence of the vertical sulci. Where we find the vertical sulci is the difference between the y, the anteroposterior y value, where we find this rostral limit of the pants, and where we find the intersection between the pax and the cingulate sulcus or the paracingulate sulcus if present. And you can see that it's tightly linked to the peak of occurrence of the paracentral sulcus occurs at the level of the rostral limit of the pons. The preparacentral sulcus, we observe the same thing, but with it's observed at the level of the anterior commissure. The posterior vertical paracingulate sulcus always falls at the caudal limit of the genu of the corpus callosum, and the anterior vertical paracingulate sulcus falls at the level of the rostral limit of the genu of the corpus callosum. So this means that the vertical sulci are located at anatomical landmarks that all primates possess. So now if we look at what's going on in the ventromedial prefrontal cortex, this region is characterized by three main sulci. The superior rostral sulcus, that seems to be a very old structure, and it's there in all primate species, in all hemispheres, in 100% of hemisphere, in all these species. Sorry. If we look at now the inferior rostral sulcus, so this is a secondary sulcus, so it's not there in all hemisphere in human. But you can see that it's present in human and sheep equally, and it's equally present in baboon and macaque, so almost no present here. So we have again this difference between hominids and old world monkeys. And the final one is the accessory sus orbitalis sulcus here, where we see a big difference between human and sheep on one side and baboon and macaque on the other side. So this means that and importantly, all these sulci in this region are sulci, even in macaques. We do not observe spur and dimple. So now, take a look at what's going on at the intersection between the dorsal and the ventral medial prefrontal cortex here. And this is the region that really display strong differences. So this region is characterized as following. In human, we have a fork that hands rostrally the cingulate sulcus. And this fork is formed by the sus orbitalis sulcus here and the suprarostral sulcus here. When a paracingulate sulcus is present, this fork is most often located at the end of the paracingulate sulcus. And this fork is always pointing downward, like this. What's going on in chimpanzee? In chimpanzee, we observe this pattern. So the fork is looking downward, that is represented here in pink, in most of the hemisphere. But in some hemisphere, about 20% here, the fork is it's forward facing like this. So now if we look at what's going on in baboon and macaque, we have more different patterns. We have this pattern where the fork is looking, is ending by a complete fork and pointing downward. And you can see the linear decrease of occurrence of this type of pattern from human to macaque. We see that the fork can be forward facing, like in chimpanzee. We can also see that in some hemisphere, the fork is incomplete with only the sus orbitalis sulcus. And in some brain, there is no fork at the end. So what we observe is that there is a gradient from macaque to human where the fork becomes more complete and the fork points more downward. And this is, we can have a look on where is exactly located this fork. And to do so, we looked at the difference between the dorsoventral Z value, where we find the apex of the genu of the corpus callosum here, and where we find the fork, and we looked at the difference. And you can see that in human, this fork is really close to the apex of the genu of the corpus callosum. But in all non-human primate, the other one, chimp, with the chimp included, this fork is located dorsally to the apex of the genu of the corpus callosum. And we do not see differences between chimp, baboon, and macaque. So this suggests that specifically in human, the fork is pushed downward. And this is concomitant with two morphological aspects. First, there is the appearance of a new sulcus in human, that is this one, the rostroperpendicular paracingulate sulcus, probably reflecting the expansion of area 9. And this is also concomitant with the full setup of the dorsomedial and ventromedial polar sulci that can be seen here, dorsomedial and ventromedial, that we think delimitate area 10. And you can see that it's there in human. It's there also, the dorsomedial polar sulcus is there in chimpanzee, but in 50% of hemisphere. And almost not, the ventromedial polar sulcus is almost not there, and it's almost absent in baboon, macaque, and baboon and macaque, both of them. So to conclude, what the analysis of sulcal patterns told us, it told us that the dorsomedial frontal cortex and the ventromedial prefrontal cortex are well preserved from macaque to human, but that major changes occur at the junction between these two regions. In addition, we have seen that the sulcal organization is tightly linked to function in the dorsomedial frontal cortex in both macaque and human. It's also the case for the ventromedial prefrontal cortex. I didn't present this data, but you can have a look on this paper to see the details. So to conclude, because sulci relate to function in both human and macaque, the anatomofunctional organization of the entire medial frontal cortex may be understood across species thanks to sulci, even in species for which we do not have MRI anatomical data. And to conclude, I would like to thank all my collaborators in US, in Canada, and in France, and my lab, obviously, the people in my lab that are really supportive, and all the grant agencies that supported this work. Thanks a lot.
Hello, my name is Olivier Coulon, and I'm very happy to present you my talk entitled Morphological Features of Sulci and Their Relationships to Function and Connectivity. First, I would like to thank the organizers for inviting me to be part of such an exciting symposium. And the central topic of this symposium is sulci. And a sulcus is a fold on the cortex, which means a piece of cortex that can be represented by the following ribbon-like objects. And this allows us to easily represent its shape, for instance, here in the case of the central sulcus. When looking at several individuals, one can see that this shape varies and has differences and commonalities across subjects. So the question is, are these shapes and their variations meaningful, and how can we characterize them? So I will start with the second question, how to characterize it. There are many ways to characterize the shape of an object, and this one is dedicated to the central sulcus. Here, you see a central sulcus, and it is possible to build a coordinate system X in the interval 0-100 that describes how to go from the dorsal to the ventral extremity of the central sulcus. And at each position, a shape feature is computed that describes how the CS, the central sulcus, deviates from its main axis in the anterior-posterior position. And we call that curve the sulcal profile. Here, you see a bend corresponding to a local minimum on the sulcal profile, followed by a bend in the other direction, corresponding to a local maximum. So this sulcal profile is defined in the 0-100 referential, and that normalizes the sulcus by its length. Therefore, it can be used to compare individuals or even species, as I will show. There is a very well-known feature of the central sulcus. It's called the hand knob, corresponding to this bend here. And using stimulation, it has been shown to localize the primary motor area of the hand in this famous paper here. So we can wonder, can we locate the hand knob in a reliable manner? So we did build an algorithm that locates the horn of the hand knob and its ventral limits under the form of two local extrema of the sulcal profile. And indeed, we showed with functional MRI that it locates a primary motor area here for, in that case, for a thumb extension task. And that resulting functional area is in between our two maxima here in the hand knob. It's a simple result, but it shows that the central sulcus morphology can be used to automatically locate a functional area. And the sulcal profile is not the only geometrical feature that can be computed. A more general one and a more meaningful one as well is the sulcal depth. Here, you can see the same central sulcus with the same coordinate system. And at each position, it is possible to compute the local depth of the sulcus, which is the distance between the surface of the cortex and the fundus of the sulcus here. And it results in a depth curve. And that allowed us to study variation of depth statistically and produce this typical here average depth curve of the central sulcus in humans. Interestingly, this curve is very similar in other species, like chimpanzees here in that curve. And you can see the equivalent curve in humans here. And actually, it's not just chimpanzees, it's all great apes. And this study here showed that human and great apes have a similar profile, a bimodal profile with a minimum in the middle. Unlike old world monkeys, those ones that all have a monomodal depth curve. So that minimum here is called the pli de passage frontoparietal moyen. And it's a French term that was first described by Broca in 1888. And I will come back to that in a few minutes. In the central sulcus, the PPFM co-localizes with the hand knob. And it was indeed shown in this paper that the pli de passage frontoparietal moyen, a depth-related feature, is a marker of the primary motor area of the hand as well. So more generally, we can wonder if the depth of fold is a feature that has any functional relevance. Well, this is the topic of this study led by Clémentine Baudin that was looking for a way to anatomically locate the temporal voice areas that respond specifically to voices in the superior temporal sulcus. For that, we built surface-based individual maps like this, and then we drew the STS fundus, and at each position of this fundus, we computed a single feature by locally integrating functional values. It results in an individual functional curve like this, with a position between 0 and 100 again, from here to here, between the two extremities. And in the same referential, we can also plot the depth curve, like here. So the first result we got is that there is a very strong and significant asymmetry of depth, with the right STS here in red being much deeper than the left one in the posterior part of the STS. This area is called the superior temporal asymmetrical pit and the STAP, and it was previously presented in this paper here by François Leroy a few years ago. So we did reproduce this finding, and functionally, we found the STAP, but functionally at the group level, the result is that in both hemispheres, here and here, the functional peak is within the STAP, shown here and here, where the STS depth reaches a peak as shown here. This is a group level result, and at the individual level, we show that functional values can predict if we are within or outside the STAP with an accuracy of 60 to 70%, depending on the hemisphere. So the STAP is a marker of the temporal voice areas at the group level. But at the individual level, this link is actually quite weak with an accuracy that is not that great. And so this is due to the fact that the superior temporal sulcus morphology, as shown here, is very variable across individuals. And its depth is also very variable, as shown here, including within the STAP in the orange box here. So how can we explain and overcome such variability? Let's go back first to this typical bimodal depth curve that I showed before. In the vast majority of subjects, the depth curve looks like this here in blue, but in less than 1% of cases, it looks like this. And these two cases correspond to such central sulcus, one piece on the left and two pieces on the right with an interruption at the level of the minimum reaching 0. So those two different configurations have different topology, but nevertheless, if you look in the depth of the central sulcus here, you can see that in both cases, you can actually see two subunits. So the difference between the two cases is that the variable depth of the PPFM sometimes reaches 0 here, and that explains the topological variation of the sulcus. The PPFM was first described by Broca and then by Cunningham and Horsley here, and they stated that it is a buried gyrus that connects the two banks of the central sulcus, and that can always be characterized by a depth variation, a distinct shallowing of the sulcus. And actually, in this very interesting paper, you can see a rare example here of a visible PPFM with an interrupted central sulcus. The concept of plis de passage is more general than for just the central sulcus, and in several papers, in particular from Michael Petrilli's lab, like those two papers, it has been used to explain variations of folding patterns. In another paper by Jean Régis, it's been the case specifically for the superior temporal sulcus. So what actually do we know about plis de passage? It's an old concept that was first introduced in 1854 by Gratiolet. It corresponds to transverse gyri that are buried inside the sulci, and they join the two banks of those sulci. And it's sometimes called annectant gyri, gyro bridges, submerged gyri. They partially explain sulcal variability, as just shown. They sometimes co-localize with function, as shown in the central sulcus. And they are sometimes associated to a specific short-range U-shaped connectivity. In the case of the central sulcus, as shown in this paper by Katja, who showed a dense and complex U-shaped fiber organization around the PPFM. And we did confirm that results with a different methodology in this study by Alexandre Pro. But the problem is that the plis de passage do lack a formal morphological definition. There are concepts that when it's buried deeply inside sulcus, is hard to detect with variation of depth alone. That's why Clémentine Baudin again proposed another morphological definition to define them. Here, you see a very visible plis de passage here in the STS. And when zooming and rotating, you can see those bridge-like features here. And for a very deep buried plis de passage, as shown here, you can see with the same visualization, residuals here of these bridge-like features. We call these wall pinches, and we propose to use them to characterize the presence of plis de passage. And given the literature, we hypothesized that this definition is associated with a specific U-shaped short-range white matter connectivity. For this, we used 100 subjects from the Human Connectome project, for which we had anatomical images and connective information, here, after reconstructing whole brain tractograms. And for each hemisphere, we located all plis de passage in the STS using geometrical criteria to locate our wall pinches, as shown here. And at the intersection of the superior and middle temporal gyri intersection with the plis de passage, we define some seeds, as shown here, seeds for superficial and deep plis de passage, but also randomly built control seeds to check the specificity of our findings. And for each pair of seeds, we counted the U-shaped fibers that go across and below the superior temporal sulcus. And we counted U-shaped fibers, or streamlines actually, from the tractogram as illustrated here. And so the results are presented here for variable seed size, because it's a parameter of the study, here for the left and right hemisphere. And the results do confirm that our morphological definition of plis de passage is associated with a specific short-range connectivity. And specifically, there is a higher U-shaped fiber connectivity under the plis de passage compared to control plis de passage here. And even more, there is a higher U-shaped connectivity under superficial plis de passage compared to deep plis de passage. So we therefore propose a morphological definition of plis de passage within the STS that is associated with a dense and specific U-shaped short-range connectivity. Here you can see a short animation that shows that indeed what we extract, the bundle we extract, are U-shaped bundles that connect the two sides of the superior temporal sulcus. Interestingly, this very good paper came out at the same time, investigating superficial white matter using Klingler dissections as shown here. And the paper focused on what they called three-ray ridge junctions that actually correspond to the wall pinches that we defined earlier. They are numbered here. And the paper confirmed their links with connectivity and showed that they are the convergence points of U-shaped intergyral fibers and also intra-gyral short-range connectivity fibers. Specifically, that paper suggests that the wall pinches are not caused by the trajectory of the U-shaped fibers, but they are probably caused by a high density of the termination of those fibers. And so being hubs of short-range connectivity and having a high density of fiber termination, maybe it suggests that those wall pinches and the associated plis de passage do have a functional role. I will finish by the following observation. Some plis de passage are common to primate species, like this one shown here under the Heschel gyrus here in the STS, can be found in macaque, baboons, or chimps. Some other plis de passage inside the STS cannot be found in other species. And so that fact should lead us to investigate the commonalities and differences of superficial white matter connectivity within sulci, and even to investigate the link, the possible link with differences and commonalities in functional organization. I will conclude with the following take-home messages. First, sulcal morphology and these variations are not random and do contain information. It can be a marker of functional areas and underlying connectivity, as I showed. The depth, in particular, is a very useful feature that has been used in many papers. Plis de passage are important sulcal features, and they are key elements for understanding sulcal variability, accessing superficial white matter scaffolding, and possibly locating function and comparing individuals as well as species. Thank you for listening to my talk, and I will finish by acknowledging the contribution of the funding bodies for those studies, and mostly by acknowledging the contribution of all the people who participated in my institute in those studies, and as well as in other teams. A lot of collaborators were involved, and I would like to thank in particular Jean Régis in Marseille and Jean-François Mangin in Paris, because they clearly are at the origin of all the work we do about sulci and plis de passage. Thank you very much for listening to me.
Hello, my name is Katja Heuer. I'm a PhD student at Max Planck Institute for Human, Cognitive and Brain Sciences in Leipzig, and I'm interested in understanding the developmental and evolutionary sources of brain organization. Today, I would like to talk to you about mechanical morphogenesis and the development and evolution of primate cortical folding. In my presentation, I will focus on the emergence of folding patterns across different primate species in the light of mechanical morphogenesis and phylogeny. The folding of the neocortex is a distinctive feature of large mammalian brains, and folding patterns are strongly conserved between individuals of one species and characteristic for each species. And the geometry corresponds in many cases with the cytoarchitectonic, connective, and functional organization of the brain. So as we can see here, for example, the primary motor cortex and somatosensory cortex to either sides of the central sulcus across the maps for human, macaque, lemur, and marmoset. And when we look across many species of the mammalian kingdom, like here, for example, in this beautiful illustration by Lea Krubitzer, we see that the size and the location of the primary of the cortical fields varies largely between species. And we can also see that the size does not increase as the cortex grows, but that more and more associative cortices are being added. And if we look across many different species, the geometric and organizational complexity vary together. So the larger the brain, the more folded the brain will be, and also the more cortical areas we will have. Now, how does the species characteristic folding patterns emerge? To what extent does mechanical morphogenesis influence the evolution of the neocortex? During the last few years, the idea that brain folding is produced by a mechanical instability triggered by neocortical expansion has been more and more recognized. However, folding patterns are still believed to be genetically encoded. And we would like to ask to what extent mechanical morphogenesis influences the organization, development, and evolution of the neocortex of the primates. A few years ago, Roberto Toro started the Brain Catalog project to study the evolution of neuroanatomical diversity across the vertebrate kingdom, together with the Natural History Museum in Paris and the Institute of Brain and Spinal Cord. And in this collection of the museum, there are many different brains of many different primates. And so we were able to study high-resolution MRI images of 34 different primate species. And we used different tools to segment the PL surface and also our collaborative web app BrainBox. And we were able to reconstruct the 3D surfaces for all these different species. And in the first analyses, we looked at the volume of the brain, the surface area, and the global gyrification. And we also developed a method to study average fold depth and width. And then we used phylogenetic trees built from genetic data and fitted different evolutionary models. And this is, for example, the simulation of a Brownian motion model, where the phenotypes are assumed to vary randomly along the phylogenetic tree. And in the next simulation, we see the Ornstein-Uhlenbeck model, where the phenotype is also assumed to vary randomly along the phylogenetic tree, but striving towards an advantageous adaptive value. And we also tested the early burst model. And among these three that we tested, the Brownian motion model fit the best to our data. And what we observed is that brain volume in several branches of the phylogenetic tree only decreased and decreased. For example, the branch leading to the Lemuridae and the Galago. In other branches, like for example, the one leading to the human, brain volume only increased and increased. And then there's branches where there is a mix of decreases and increases of brain volume, for example, in the branch leading to the tufted capuchin. And these phylogenetic models also allow us to estimate the ancestral traits along the phylogenetic tree. And the brain of the common ancestor of all primates about 74 million years ago has been estimated to have a brain size and degree of folding comparable to the brain of an aye. And interestingly, we also observed a strong conservation of the fold width, which is very close to 1 centimeter across all these different primate species, starting from the vervet monkey all the way to the humans. So despite a more than 20-fold difference in volume, we observe a very conserved brain folding width. And so to conclude, this analysis has shown that there is an interesting nonlinear relationship between volume and wavelength, and also that random changes may be an important force for primate brain evolution. Now, how do these folding patterns emerge? Do they result from specific genetic programs? If folding patterns resulted from genetic programs, we should expect that primates that are closer in the phylogenetic tree would have a folding pattern that would be more similar than compared to primates in farther apart branches of the phylogenetic tree. And here I highlighted a few that we will look at particularly, and you see at the different colors already that they come from different branches of the phylogenetic tree. For example, the crab-eating macaque and tufted capuchin. We developed a method that allows us to encode folding patterns as a graph. So we start from the 3D surface reconstruction, we remove all the sulcal regions, and then we are able to shrink this gyral Gruyère into a wireframe, which we can then represent as a graph. An adjacency matrix where the edges are the gyri and the nodes are where the gyri fuse, and the holes are the salci, which we annotated here, a few of them, so for better orientation. And this is, for example, the fold graph of the tufted capuchin. And here you can see for different species of our collection, the fold graphs and appreciate the display of their complexity. And we use the graph at a distance algorithm to measure how similar these graphs are. So the graph at a distance algorithm allows us to estimate the number of edits we would need to derive one graph into another graph. And here we did that for the example of the tufted capuchin and crab-eating macaque, for example. If you look at the phylogenetic tree, you would expect that they wouldn't have a similar folding pattern, more similar than the duoculi, for example, which is very close in the phylogenetic tree, where you would expect a similar folding pattern. And for the crab-eating macaque, the common ancestor has been 47 million years ago. And what we actually see with the graph at a distance measurement is that to derive a crab-eating macaque into a tufted capuchin, only a very few edits are needed. However, if you want to transform a capuchin into a duoculi, which are phylogenetically closer, the folding pattern is much more different. And so that's very interesting because the tufted capuchin is a particularly interesting example. Its ancestor may likely have had an unfolded lissencephalic small brain about 47 million years ago, but then the tufted capuchin derived a larger brain and ended up very close in terms of brain volume to the crab-eating macaque. And we can see that this is not just an outlier. We see it across many different species. So here, for example, king corvus, white-faced sapaju, gray-cheeked mangabey, and many of them have a common ancestor 47 million years ago, likely lissencephalic and small brain. And here, more than 20 million years ago, the common ancestor. And yet, just based on a more similar brain volume, these species have derived a very similar folding pattern. So these analyses suggest that mechanical factors may play an important role in the evolution of brain organization, and that folding patterns are more similar given a brain volume similarity rather than the position in the phylogenetic tree. So if folding patterns were genetically encoded, tufted capuchins should have evolved in parallel a similar folding pattern to the crab-eating macaque. It's more likely, however, that folding patterns, same as degree of folding, are constrained by mechanical morphogenetic forces. And it has been shown that the initial geometry of the unfolded cortex may be a strong determinant of the orientation of the folding. In addition to the initial geometry, our global shape, also global expansion gradients should influence mechanical morphogenetic processes, which then lead to the formation of stable folding patterns. And we recently studied these expansion gradients using a nonlinear surface deformation algorithm to create homologies between all species. And this resulting evolutionary expansion trajectory going from the small lemurs toward the great apes illustrates a striking continuity, such that folding patterns were similar along species with comparable brain volume, despite their position in the phylogenetic tree, underlining again the importance of mechanical morphogenetic processes in the definition of folding patterns. And to better understand this evolutionary trajectory, we fit a surface-based Gompertz model, which allowed us to map differences in expansion rate and expansion onset. And we see that the evolutionary trajectory we obtained showed a caudal rostral gradient, where the more caudal regions start expanding earlier and faster than the more rostral ones, which start to expand later and take longer. So to conclude, given a neocortical thickness is very stable across primates, mechanical morphogenetic processes should lead to the formation of folds of similar wavelength, organized in a pattern influenced by global shape and global expansion gradients. And within this framework, brain folds would act as mechanically canalized, strongly polygenic modules with stereotypical shape and large-scale gradients of mechanical stress, which could then influence cell proliferation, cell fate, and connectivity during development and evolution. And with that, I would like to thank all the people that I have the pleasure to work with, and especially Roberto Turo in Paris, who is working with me in all these projects, and Nicolas Trou, who joined our team as well. Thank you very much for your attention, and I'm looking forward to our discussion.